Hello, this module is on translation, but specifically on initiation. So we're going to look at how Nirenberg dissected initiation, actually Nirenberg and others, how they dissected the initiation process. And Nirenberg assumed from his prior experiments that there was an association of his ribosomes with messenger RNA. You'll recall that that's in fact the basis on which he did experiments that eventually deciphered the entire genetic code. So let's look at initiation. It turns out that messenger RNA actually binds to the 30S subunit of the ribosome. So here we have a sucrose density gradient. Remember that we use sucrose density gradients to separate different size classes of ribosomal RNAs, or whole cell RNAs, which turned out to be ribosomal RNAs for the most part. We did this on a sucrose density gradient. Well, you can also separate particles on a sucrose density gradient. Now, particles, of course, are much bigger than molecules of RNA. So if I separated molecules of RNA on a gradient, say, that went from, say, 5% to 20% sucrose, if I want to separate particles, which are much larger and more massive, like ribosomal subunits, I'll centrifuge them in an ultracentrifuge on a sucrose density gradient that might go from, say, 10 to 40%. Remember how soluble sucrose is in water or in buffers for this purpose. So we have separated here large and small subunits. The two bands, these two brown bands, are different thicknesses because there's going to be physically more of the large subunit material than of the small, even though there is one each in a ribosome, right? Because the large subunit simply has more stuff in it. So there are our two bands, and we can fractionate them, and you can see that the lower band is the bacterial 50S ribosomal subunit, and the upper band, the one that moved more slowly in the gradient, is the smaller 30S subunit. Well, one can do simple experiments. Add a messenger RNA, perhaps one that's been made radioactive so you can track it, and add, say, cytosol from E. coli cells, and ask if there is an association, and it turns out there isn't one. On the other hand, if you add the 30S subunit, the small subunit, to a radioactive mRNA with cytosol present, presumably to provide some necessary factors for things to happen, sure enough, the 30S subunit can be shown to associate with the mRNA. How do we know that this association is actually taking place? How do you show this? Well, very simply, you take this material and you run it back on a gradient, and you show that the 30S subunit, where it fractionates in the upper part of the gradient, is now radioactive because it's had mRNA added to it. It might even be moving a little faster because the mRNA adds some bulk to this structure. So here we have a picture of the sucrose gradient that shows that the radioactive complex would be larger than the mRNA alone on a sucrose gradient, and in fact would be about the size of the 30S subunit. If the two are mixed, then they come together. So what is the role of the cytosol? I keep talking about adding cytosol here to make things happen. So what is the role of the cytosol? Well, it turns out that cytosol contains initiation factors identified as cytosol components. There are actually several, and they are understood in some detail. So if we mix 30S subunit extracts, isolates, with initiation factors, or IFs, isolated from cytosol, with a real messenger RNA that's been made radioactive so we can follow what's going on, we get this association. So what do these initiation factors do? Well, one of them enables the 30S to actually bind to the messenger RNA, and the binding is H bonding of the mRNA, the beginning of the mRNA, a sequence called the Shine-Delgarno sequence, discovered by these two guys, near the 5' end of a message, actually recognizes complementary bases in the 16S ribosomal RNA of a bacterial small subunit. Initiation factors facilitate that interaction. Some initiation factors are actually there to prevent the 50S subunit from binding, and you'll see why that's necessary in just a second. So we're still in initiation. What happens next? One possibility then is that the 50S subunit simply attaches to this complex, but as you see, that doesn't happen. So one of the initiation factors seems to be responsible for preventing a 50S association with this structure. Well, we're concluding here that the large subunit does not yet combine with the mRNA. You know that it's going to have to, but it doesn't do it yet. It doesn't bind with the mRNA small subunit complex. Well, we know that amino acid tRNAs are going to have to become part of a translation structure, so perhaps that's the next thing to happen. People had analyzed or extracted E. coli polypeptides in the 50s and 60s, and in the 50s was the first time that people had begun to actually do amino acid sequencing. It was not automated, it was very tedious. But amino acid sequencing of a number of E. coli polypeptides, enzymes, and other proteins revealed that of the ones that were sequenced, 40%, which is a large number, 40% of these all had methionine at the amino terminus, at the end terminus of the polypeptide. You'll remember from our discussion of protein structure that polypeptide chains have an amino and a carboxyl terminus. Well, methionine was found at the amino terminus of about 40% of E. coli proteins. Now, you may also recall from the genetic code dictionary, because I pointed it out to you, that there is one codon for the amino acid methionine. And this is important for understanding the next bit of investigation. Several researchers found that, in fact, there were 2 transfer RNAs for methionine. You would have predicted that there would only be one. Since you have only one codon, then you would only need one methionine tRNA with its appropriate anticodon. But 2 of them were found. They were met tRNA sub-met, meaning they were found with methionine on them, so they were methionyl tRNA sub-methionine, meaning the tRNA for a methionine. And then there was one with a methionine on it, all right. It was a methional tRNA, but it had an organic group on it, a formal group, F-O-R-M-Y-L, formal group, which was part of the methionine. And so the tRNA then was called tRNA subformal met, or fmet for short. So these were the 2 tRNAs with their versions of methionine attached that were actually found in E. coli cells. Here we have them drawn out. I'm showing methionine here with a formal group circled on the formal methionine. And these were then, of course, attached, or acylated, we say, attached to the tRNAs, to their respective tRNAs. So it turns out that the tRNAs were isolated, purified, and actually sequenced. tRNAs are relatively short. They're 70 to 80 or so bases long, and were the first nucleic acids to actually be sequenced. Nowadays, DNA and sequencing is routine, RNA sequencing less so, but not impossible. But in the 60s, mid-60s, it was quite a feat to sequence actual RNA molecules, and the tRNAs were sequenced in part because they were short, and in part because they have a lot of base modifications which made the chemical sequencing of these things a bit easier. So you may remember, tRNAs are processed after they're transcribed to include a lot of unusual bases. So they could be sequenced, these 2 tRNAs, tRNA sub-met and tRNA sub-fmet, and they had different base sequences, but they did indeed have the same anticodon. So the question was, what is fmet for? Why do you even have this? Why are there 2 tRNAs, and why is there an fmet? There were some speculations, and one was the formal group, this CHO, is blocking the amino group of the methionine on which it is placed. And what that might have done in evolution would be to prevent the formation of a peptide bond, to prevent a condensation reaction of another amino acid to the N-terminus, to the amino terminus of this formulated methionine. In other words, it might restrict chain growth in one direction, so that the chains would grow by addition to normal-looking carboxyl ends of amino acids, starting with fmet. So it was not possible, if you started with fmet, to then add amino acids to the amino terminus of this obviously different methionine. Well, that's a reasonable speculation, and in fact, in the mid-50s, experiments had been done to show that indeed chain elongation during polypeptide synthesis does indeed proceed by adding new amino acids to the carboxyl end, never to the amino end. So amino acids grow by adding in one direction, N-terminus to C-terminus. So the only purpose that the fmet tRNA could reasonably serve is to be the first in a polypeptide chain, the first one to associate with a 30S subunit that is now stuck on a messenger RNA, perhaps with a few initiation factors. There is an enzyme in E. coli which has been isolated, which has the ability to recognize methionine when it's bound to the fmet tRNA sub fmet, and that formulating enzyme catalyzes putting a formal group on the amino end of methionine. That same enzyme is unable to recognize the methionine bound to a tRNA submet, which explains why one methionine gets a formal group but the other one doesn't. So that's the role of the formulating enzyme. So as it says here, formal methionine tRNA fmet, there's one T missing I see here, but that's okay. Formal methionine tRNA fmet is indeed the first in translation, and here's how it happens. That association requires another initiation factor, so we've added another initiation factor to the IFs shown here, and it requires GTP as well. And it turns out, of course, when you do this sort of experiment, no other amino acyl tRNA does this. So here we have the same sort of thing happening again, but with a bit more detail. It turns out that if you do these experiments, the GTP and the fmet combine with the 30S subunit and the initiation factors that are present to form an initiation complex. Now let's look at the completion of the assembly of a ribosome. Let's finish assembling this ribosome. Well, it turns out it's very easy. Now, just add the 50S subunit, and this was, this sort of thing was done in test tubes in step-by-step fashion. All of these things I'm showing you were extremely elegant molecular experiments in which different parts of the cell had been isolated, kept in separate tubes, and then the order of events established by carefully adding one thing to another and then tracking changes. So here, all you had to do, once you had the initiation complex, was add the 50S subunit, and the GTP was hydrolyzed, the GDP dissociated from this structure, and all of the initiation factors also dissociated from the structure. So that brings us to the end of this presentation.